So, right, last time I was talking about a lot of biology about the worm, this nematode worm C. elegans that my lab studies, and we're getting kind of really specific into a certain protein, PPH4, and how it's involved in multiple steps in meiotic protease. Mostly I was doing that not so you learn about PPH4 itself, but just as a way to illustrate what those steps of meiosis are. And at last lecture I was talking about the final step that PPH4 is involved in, which is recombination and crossing over. And without PPH4 function, these steps of recombination and crossing over also fail, so we think it's required to do those steps properly. And you probably remember I showed some diagram kind of very quickly and cursorily like this, showing some DNA strands and some DNA invading other DNA. And I said I would get back to it later in another class. So that's what today is going to be about. We're still not going to go over every single step because there's more here than is really important for meiosis, but most of this we're going to at least take a look at. And so I should say that this area of DNA recombination is really important, and it's really it's a field that's been around for a very long time. Since the very beginnings of molecular biology, people studied DNA recombination in bacteria and viruses. When people were first figuring out what DNA was, one of the first things that was discovered was that all DNA likes to switch pieces of itself with other DNA molecules. And the reason it does is because one piece of DNA by itself will eventually degrade and lose all its information over time. So DNA always has to be repaired. And the way DNA repairs itself is by doing this process called recombination. So when we're talking about DNA recombination in the context of chromosomes, and especially in meiosis, there are a few terms that we have to get clear on, especially when you see a picture. When I draw a picture, sometimes I'm just going to draw a line, or you'll see a diagram with just a line. And the first thing is very important to always know what you're looking at. So am I drawing a single strand of DNA, so a single 5' to 3' DNA strand, or does a line mean this is a sister chromatid, or does a line mean this is an entire paired or unpaired homologous chromosome? So I don't think I will use a color code or anything to keep track. But if I ever do this, if I ever draw a line with a 5' at one end, then you know that this is a single strand of DNA, and it's usually going to have a 3' partner, and then it will end like that. So if I draw both of those, then this is a double-stranded DNA. But then sometimes, if I'm drawing a crossover, I will draw something like this. And so here, it's quite confusing when we get ahead and look at things like this, because this is just this is a single DNA strand. But when I'm drawing like this in the context of meiosis, this is an entire replicated chromosome. So this is chromosome 1 from the father in 2 pairs, and then this is chromosome 1 from the mother, and it's also replicated. So this is an entire replicated chromosome, and then this is a pair of homologs. But you'll note here I'm not drawing 5, 3, 3, 5, because if I were, I would have to draw like this: 5, 3, 3, 5 on the top, and then 5, 3, 3, 5 on the bottom. So in other words, what's here on top is one chromosome from the father, but there's actually 4 single DNA strands. And then on the chromosome from the mother, there's also 4 single DNA strands. So what's going on here in a crossover is not the same thing that is going on here with recombination. I'll try to point out what I'm doing as I draw it or as I talk about it, but if you have any questions about what you're actually looking at, it's important that we clarify that as soon as possible. So when we say recombination, we're talking about one DNA strand switching with another DNA strand. And we're talking about crossing over. That means one chromosome is switching within another entire chromosome. I think we all have in mind meiosis 1 and meiosis 2, and that's also something to just keep in your mind when you see chromosomes coming apart. Just make sure you know what you're looking at, whether this is the first or the second meiotic division. And in the first meiotic division, of course, we know that homologs stay paired with each other, but they in the second meiosis division, it's just like mitosis when all sister chromatid cohesion is removed. Okay, so I've said many times we're gonna go ahead and look at recombination. So let's get started with that and see what actually happens when the chromosomes are together. So it starts with we have chromosome. Let's just be stereotypical and say that the pink chromosome is from the mother, and the green chromosome is from the father. And these, let's say that they're paired together in the context of the synaptonemal complex. And this is when the SC or the synaptonemal complex is between them. This is when recombination is happening. So previously during S phase, of course, this chromosome is replicated, and the father's chromosome is also replicated. So now that these two replicated homologs have come together, they're in a position to be able to recombine. But you'll notice, as I draw them like this, the DNA is actually not touching each other yet. This DNA is over here, and this DNA is over here. But I'm not showing you, or this is just a diagrammatic way of showing it. But remember, these DNAs, they really exist as these loops, and these loops are going all over the place. So when we think about recombination happening, it's happening between DNA that's touching at these loops inside this chromosome. Okay, so this chromosome is over here, and this chromosome is here. But let's now focus in on what's happening at the DNA level. So now I'm going to just focus on one DNA molecule from each parent. And let's say they've touched each other up here. So we're not talking anymore about the main axis of the chromosome that's away from the other one. We're talking about the stuff that's happening maybe right here at that intersection. So first of all, for recombination to happen, this DNA has to be allelic, which means that the sequence has to be the same on the top and bottom strands. You'll note that I drew these strands flipped. So here I have a 5 prime. The top strand is going 5 prime to 3 prime, and the bottom is going 3 prime to 5 prime, whereas on the bottom strand that's reversed. And that's perfectly okay to do, of course, because if you remember, DNA is helix. So it's really they're both helices, they're both identical. So that's this is just to make it easier to see what actually happens. So let's just assume that this part of the chromosome wants to recombine. We won't go into why yet, but it's decided that this is where recombination is going to happen. So the very first thing that has to occur in order to get recombination is you need to first break the DNA. So I said last time that cells normally are really paranoid about breaking their DNA. They want to avoid breaking DNA at all costs. But in meiosis or during recombination, DNA gets broken. It has to be broken. So in meiosis, there's an enzyme called SCO11 that comes along and specifically, intentionally, well, as intentionally as you can be in biology, creates a double-strand break in the DNA. So, and the thing about a double-strand break is that once it's done, and usually it doesn't stop there. There's a bit of a few bases are missing there. Now this information is gone. Whatever was here at this locus on this chromosome is gone. And now that the the chromosome has no choice but to repair it somehow. If at the very last ditch effort in a cell that's not going to go on to reproduce, sometimes they just put the DNA, they just put it back together. Because that's better. This gene might mutate, you might lose some information here, but if you don't do that, of course, you've broken the entire chromosome, because this little break here is actually enough to break the entire chromosome, and you will have 2 chromosomes there, only one of which will have a centromere, and then you'll lose the other part. So you absolutely have to repair this. But a better way to repair it, especially since you're here in meiosis and you know that you've paired with this partner down here, is to use the information that's down here to repair what's up here. So in order to do that, you first have to expose part of this DNA strand so that you'll be able to find the right information from the bottom strand. So you can see that what happens is this, I hope you can read this, it says 5' to 3' resection. So each of these steps, every time you see an arrow, what's actually happening is that one or more proteins are coming along and doing this step. So this diagram doesn't say which proteins are doing it, and that's just fine. It would be too busy and too hard to remember all of those proteins. But there are proteins that come along and recognize this kind of break and say, okay, I know what to do next. Now I'm going to go and degrade even more. So the first thing to do, it's kind of paradoxical, but the first thing the cell does to repair damage like this is it makes even more damage. Okay, it starts doing this thing called resection, which is just chewing back these bases. So whatever bases were here on this strand are just cut off, thrown away. The reason this is done is because is to produce this single-stranded region of DNA. So now you have this, so the DNA has been broken, and now you've got this single-stranded region, which actually, I'm drawing a kind of small here, but it can be 100 or up to maybe even 400 bases. So that's long enough to go to search quite a large area around it. And it can search and search until it finds this homologous partner, right? And that's what's happening right here. This extra little tail of single-stranded DNA is able to come in here and invade this other strand. And you can see here that there are 2 proteins mentioned. One of them is the RAD51 protein that I mentioned last week. That's a protein that makes this kind of, it itself makes a helical wrapping around this single-stranded DNA. And the combination of RAD51 plus single-stranded DNA is powerful enough to displace this strand and come in and base pair with the strand that's down here. So now the single-stranded part that's been exposed by resection has come in, and it is actually, so the base pairing is maintained the entire way. I've only drawn 3, but it's actually a few hundred base pairs that are right there. So what's the point of doing this? So the point is that once you have, once you've made this connection, remember you needed to repair this gap here. And so, okay, so now there's 2 ways you can go. There's actually 2 ways you can repair it. We'll start with the one that has the most importance for meiosis, and that is you get new DNA synthesis wherever you have a free end. Okay, so when we talked about DNA synthesis earlier, remember that synthesis of DNA always starts from the 5' end and goes toward the 3' end. So there's 2 places here that you'll be able to synthesize new DNA. One of them is here, right? this is a free end. This is just the end of that DNA that's been exposed. And so DNA polymerase can come and start synthesizing DNA along here. So as DNA polymerase continues and adds more bases here, it keeps pushing up the other strand. It keeps displacing it. So this bubble of DNA keeps getting bigger and bigger. Eventually, it becomes big enough so that it is now able to match the other end of DNA that's still hanging out here waiting for this event to happen. And then once this bubble has become big enough and can bind to this other broken end, now this template, this end becomes competent. You've got a template here, you've got information here that you can synthesize more DNA from. So that's how you get new DNA synthesis that covers the entire gap. So where before you had this large gap of information, these are precisely the places that you have made new DNA synthesis. All right. So there's another way to do that. Of course, you didn't have to go ahead and do any base pairing with this D loop. So this bubble doesn't actually have to trigger new synthesis of that strand. It's totally sufficient to just repair one strand, right? As you can see, if you just repair even that one strand, it can pop out of this bubble and then serve as a template to repair the top strand. So this is kind of an escape mechanism, and this actually happens quite a lot in meiosis as well. But you'll note that at the end, what happens is these two chromosomes they stay apart. Okay, so this DNA molecule and this DNA molecule are no longer linked together. Maybe a little bit of information got copied from the bottom strand to the top strand, but these chromosomes are not connected anymore. But it's the job of meiosis not only to repair this damage. In fact, if we go back and think about why we were doing this in the first place, why did the cell want to make DNA damage only to repair it? That doesn't really make sense. But the point really is that you need to make connections between the chromosomes that will hold them together until cell division. And in order to do that, you have to undergo this pathway where the DNA unwinds and make the bottom and the top DNA strands make base pairing with each other. Okay, so now we have entered onto this, we've made this structure. Once you've synthesized DNA on both ends, I should say after that synthesis happens, so we've synthesized DNA to here and synthesized DNA all the way up to here. But we still have these broken ends, right? This end hasn't connected to that end yet. But it turns out that another protein comes along and does that job. So another protein called DNA ligase. Ligase just needs to tie it together. Ligase comes along and makes this representative dotted line into an actually solid line. So in the end, what you really have are connected continuous DNA strands that that there's no gaps anymore, all right? And it doesn't really look like this. We need to remember, I'm going to go through, so I've just shown you this very traditional way of drawing things with these lines. But we need to keep in mind that DNA is a right-handed helix, and that introduces this kind of three-dimensional geometry aspect to this whole process. And I find that visualizing it this way makes it a little, maybe even a bit easier to understand. So I hope you can see, I tried my best to make these right-handed helices, although they're kind of, it's a little more angular, and there's no minor or major grooves here. But just think of this, these are DNA molecules. And the top and bottom one would be the paternal and maternal chromosomes again that are going to recombine. So just like last time, We're just looking at a small region here where the DNA is in sync with each other. So the first thing is that spool 11 comes along and cuts the DNA. And I'm showing 2 molecules of spool 11, and that is actually how it works. So spool 11 comes on as a dimer, and it cuts very symmetrically. That's how it can cut the top and the bottom strand, where it cuts both strands with a certain spacing. There's this little bit of offset spacing. Okay, so after the double strand break is made, then there's this bit of missing information that has to be repaired, or else you may get a mutation. So this 5' to 3' resection that I drew earlier just by erasing it. So this is now what that really, it looks like this, a little bit, this is a little bit better way of seeing it. So you can see these single-stranded tails. One is light blue and one is dark blue. I hope you can see that. And so this D-loop formation is not just coming in, but there's actually this invading strand actually comes in and it makes this it makes a normal DNA double helix with the bottom strand. So this is kind of a 2-frame animation of that happening. So then that continues, right? So the bottom helix keeps going as DNA polymerase synthesizes it. It's actually keeping going, winding around and displacing the other one, and finally displaces it long enough so that it starts doing again the wrapping around the top strand. So these strands aren't just touching each other. Remember, they're wrapping around each other like that. And then eventually you get this structure after everything has been ligated together, called a double Holiday junction. And so we need to, there's a few things we need to see about this double Holiday junction. First, well, what's a single Holiday junction? Well, this is a single Holiday junction. A Holiday junction is a structure that was theorized to exist and then shown to exist with electron microscopy by a guy named Holiday. And it's simply a place where 4 DNA strands come together in a perfectly kind of rotationally symmetrical structure. So when you draw it, when you draw it flattened out like this, it's hard to make it look really, really symmetrical. Anyway, these 4 strands are all actually equivalent. So I've shown 2 of them coming in this way and 2 coming in that way. But in reality, as you'll see, they're actually they come together, and actually it's a tetrahedral structure. So they're really kind of coming in to one place from different corners of what would be a 4-sided pyramid like this. So here's the Holiday junction, and each DNA strand is coming in at an angle like that. So that's what they really are like in 3 dimensions. So you can think of these as the kind of centers of 2 tetrahedrons. So to see how this Holiday junction, what the next step is in recombination after you get this double Holiday junction structure, we need to put this through one more little change. So imagine that we're taking these ends, so the ends of the green chromosomes, and just kind of wrapping them into the middle of this. So we're just sort of twisting around each Holiday junction. We're not actually changing anything, we're breaking any DNA, but we're going to isomerize, or just change the shape of this into something equivalent, which is that. Okay. So let's go back and forth a few times. So what you have here, you've got a blue and a green DNA molecule coming into one Holiday junction on the left. Okay. And you still have that here. So we have a blue and a green chromosome coming into a Holiday junction on the left. And then you have, on the right, you've got a blue and a green departing that, a different Holiday junction, and you still have that on the right. And then what you have in between them are, if you look closely, you'll see that there's actually a heteroduplex region. And that's an important term here. Heteroduplex, you'll see it's made of both a blue strand from this top chromosome and a green strand from the bottom chromosome. Because remember, when one strand invaded the other strand, you got base pairing between one and the other. And I said that these chromosomes had to be allelic. The sequence had to be the same on the top and bottom. But in practice, it's not always 100% identical. There could be a few bases different. And that's important for the outcomes of meiosis. So even though DNA is normally 100% complementary, if you have a G on one strand, you have a C on the other strand, you have an A on one, you have a T on the other. In regions like this, where you have had this migration of an invading DNA strand, it's possible to have non-homologous or non-complementary DNA sequences in there. So anyway, I hope you can see that you're kind of opening up this region here, and then putting these two green arms in the middle. So see, the green arms are going in the middle. You see how? Okay. I hope it still takes me a while. If I try to draw it from scratch, it takes me a long time to remember how this works. But I hope what you can see is now that it's flattened out, you can see that these holiday junctions are actually completely symmetrical, and there's no way for any enzyme or anything in the cell to tell, for instance, that this DNA coming in here is different from this DNA coming in here. Structurally, each part of this holiday junction is identical. You may think, okay, so this is what's happening right here. You're getting this holiday junction right here, this place where the DNA meets. Well, what about the rest of the chromosome? How does that work out? So the rest of the chromosome keeps going like this. So the maternal and paternal chromosomes are still, they just continue out from this holiday junction like this. So in this little bubble, one of these strands has to be going through it, but it really doesn't alter the structure of the entire chromosome that much. So this is what happens. I hope you can kind of imagine this is the three-dimensional structure of what is typically drawn in a textbook as this, as a double holiday junction. And I think it's much easier to see what's going on if we think about it like this, in a three-dimensional structure where each of these holiday junctions is symmetrical. It's four-way rotationally symmetrical. So I should just say that a lot of this stuff has been figured out, and there's a professor, he's a friend of mine, he's retired now, but he's from in Massachusetts. He gives a very good lecture on this whole process, much better and more clear than mine, I'm afraid. But you can, if you're curious about learning a little more in depth, this is the title of his lecture, and if you search this online, you can see him give about an hour lecture on this. But mainly I'm showing not only for that, but also for what the electron micrograph image looks like of an actual single holiday junction. This is just one holiday junction here. But these are four double-stranded DNA molecules meeting here at the middle, and then they're connected also with double-stranded DNA. Okay, so now what happens next? So I think I need to back up a little, because here's a point where it's easy to confuse what I'm showing you here with crossing over between chromosomes. So this is the double holiday junction. So what does that look like? Let's say this is the maternal chromosome, and then this is the paternal chromosome. So that's the crossover. So remember when I was showing you, you could get this double crossover in genes. Let's say this is the round gene, and I forget the actual order, the yellow gene, and maybe the pea pod color gene. And this would be an example of a double crossover between these two chromosomes, right? So let's say you started off with a male that was RYP, and crossed with this female that was RYP, and in the next generation you got an individual that was small r, big Y, small p, like that. So to explain this sort of, to explain this kind of genotype, you would have to say there was a double crossover, right? So my question is, are these two things the same? If you have a double crossover between chromosomes, is that the same as a double holiday junction between DNA molecules? Yeah, this is where it's kind of tricky. Anybody have anything, or just wait for me to explain? So yeah, this is where it's tricky, because as I've drawn them, they look identical if you just look at these lines. But what's really going on is that this double holiday junction is really just happening right here, a single crossover. So in other words, what I've drawn here these two things are the same, right? So in other words, in order to get a crossover between chromosomes, between the maternal and the paternal chromosome, in order for them to exchange DNA, you need to start with this double crossover intermediate. Okay, so the strands of DNA need to exchange in two places in order to exchange the chromosomes once. And it kind of makes sense because you have two strands that you need to exchange in total, right? So there needs to be two places where those strands get cut. But let's go into a little more detail about what happens next, and then we'll understand that part. So we have this double holiday junction. So if you think about it, you've got this. You have one chromosome coming in from the left. And then it departs on the right. So that's this guy here, coming in from the left and departing on the right. And then on the bottom, we've got this chromosome coming in from the left and departing on the right. So sorry the colors are different. So the green is the same, but think of this pink here as a blue one. I guess I should have used blue for this, but I wanted to maximize color contrast here. So the green one in both cases, let's say, is the male. And what we want as the outcome, what we want to have is one chromosome that starts as the female chromosome and ends up as the male chromosome. In other words, we want to start blue and end up green. So it seems like we should be able to do that because you said this structure here is completely equivalent. So in a way, when a DNA double helix comes into this double holiday junction, now it's got a choice to make. It can continue that way or continue that way, and those are really equivalent choices to make. So what we want is, for this, we want to come in here and end up coming over here, right? And that will be how we join this end to this end, is by taking one of these branches. Or we can take this bottom branch here like that. Okay? But either way, we need to cut some DNA for this to happen. So for this next step, another enzyme comes along and does that job. This enzyme is actually not just one, it's a whole complex of enzymes called resolvases because they resolve these four-stranded structures called holiday junctions. And the cool thing about these resolvases is it's still a little bit controversial in the field, but the model right now is that the resolvase sitting on one holiday junction does not really know what the other resolvase is doing. So each resolvase has the option of cutting these strands either this way or this way. So what I'm showing you here is a case where the left resolvase has cut these two strands like this. Okay, and the resolvase on the right has cut these two strands like this. So if you can follow now the path of how the DNA will go after those cuts have been repaired. So in this case, what happens after cutting is, again, this DNA ligase comes along, and instead of maintaining this holiday junction, it simply repairs it. It repairs this single strand together, and this is what you get. You get this this product called a non-crossover. So we didn't succeed in crossing over this time, the way we cut these strands. So we start off blue. We have this little bit area of heteroduplex, but then we're still blue. So we didn't change DNA molecules. We only picked up a little bit of information. And if we now spread the DNA back out, this is what it looks like. So what happened? These resolvases, since they cut this way, they cut in this fashion, that did not lead to a crossover. Okay. So let's try that again. Instead of cutting that way, let's switch it and cut that way. All right. I hope you can see, though, of course, the outcome is still the same. So we just took a different branch, but still we start with blue and end up with blue. The only difference is that the heteroduplex region is different now. So there's still the potential for there to be some difference in those strands. So we can still say there's been some recombination, but we didn't cross over. So now that we've been going through this, how do we have to cut these strands in order to? So at first we were cutting it like this, first like this, and that didn't work, and then we cut it like that. So in parallel. Yeah, right. So we have to cut it like this or like that. So in other words, the two resolvases need to be cutting in the same sense, in parallel, right? So now, and again, these red things are the enzymes, and they don't know what the other one is doing. They just all they do is they cut strands that are opposite from each other. I guess they never cut strands that are like this. They never will cut that and that, because that would just chop off this piece of DNA and the rest of it would fall apart. So they're smart enough never to do that. They've only cut across like that. But now, I hope you can see that if you cut like this and then just repair, then you'll get this sort of crossover. So you'll start with blue and then end up with green. So that's what has to happen in order to get this. So this crossover here is actually the end result of resolving this structure. And of course, if you cut in the other sense, you get the other kind of crossover. And again, those two crossovers 1 and 2 are only different in which part of heteroduplex that they have. And to see it a little bit close up, we can see now, yeah, I made a little bit better diagram. But somehow I couldn't, my drawing skills are not good enough to get it all correct. So don't pay too much attention to this. But this is kind of better scale model of what's really happening. And again, because the whole chromosome is still going, this is another look at what is actually happening when those things get resolved. To get a crossover, then this is what the actual resolution looks like. So we went from this double holiday junction where everything is symmetrical and anything can happen, And just by making 2 cuts, now you can see how this gets resolved. So one chromosome comes in, and then it's green when it exits. There's just this extra little loop where the DNA switched strands. But this loop quickly kind of gets resolved, and you don't see it as a loop anymore. And that's the other way that could happen. So sorry, this is how you can get a non-crossover. Okay, so this is now, this is what this heteroduplex region. I mentioned that when you have this crossing over, so if you do or do not have crossing over, you will always get this little region where one piece of DNA invaded the other piece, and whatever differences there were between the top and bottom strands remained there. Even if there's no crossing over, you can still pick up, so some information can be exchanged on one of the 2 strands. And that happens both in non-crossover cases and in crossover cases. So when you get a crossover, you can still have some heteroduplex regions where the DNA is not necessarily the same. So something that I said at the very beginning, something we need to keep in mind, is the difference between recombination and crossing over. So even in a case like this, where there's been no crossing over, there's still been recombination. So anytime part of the DNA molecule is cut and joins another part of the DNA molecule, that's recombination. So it's important to keep those 2 things separate in your mind. So you can have recombination without crossing over. And in fact, that this other escape mechanism where you don't even make this holiday junction, you just get a little bit of DNA and come back. There's still been recombination in that case as well. So recombination is always the outcome of cutting DNA, unless you get this emergency mechanism of just sticking the ends back together. Okay. So now let's just look a little bit about what happens to that heteroduplex DNA and what effect that can have later. So after the first division, of course, these 2 chromosomes come apart. So this is the meiosis 1 division. And then during the second division, the sister chromatids have come apart. Okay, so let's just focus on the top one. So this is just to be clear, these are DNA strands. I didn't write 5 and 3 here, but this is the 5' to 3' strand on top and the 3' to 5' strand on bottom. So this this first division, this is actually just replicated chromosomes. These are not 2 homologs together. I have to keep in mind that ones are too. But let's close up on the heteroduplex region. So what happens at the next mitosis? So now after meiosis 2 is finished, now this DNA is all by itself in the nucleus. It's a single piece of DNA, so it gets replicated as well. And now when it gets replicated, so the DNA unwinds, and now there's new DNA replication. And now finally there's no more heteroduplex, so now everything is guaranteed to be complementary again. But it might not have been before, and that's the point. So there may have been some mutation that was on one of those strands, and now that mutation is only on one of these 2 daughter cells. So finally, the possible products that you get of meiosis, it's more than actually 4. There's actually 8 possible products of any meiosis. Considering one place where you recombine. So you can have, of course, the most trivial one are these non-crossover events. So at the very beginning here, I showed you that all the recombination is happening between this strand and this strand. So this other chromosome did not even cross over, and this bottom chromosome did not cross over at all. So these non-crossover chromosomes are still there, and they did not undergo any recombination. But that's fine, because only one of the two sister chromatids needs to undergo recombination in order to keep the chromosomes together. So yeah, really these two are always going to be identical, and these two are always going to be identical. But between the two, between these four crossover products, they will be slightly different. The green and the blue will have slightly different extents. And that can be very interesting just in the case where one of your mutations lies in this region. If that's the case, then your products of meiosis II, for instance, one single sperm cell may give rise to a mosaic organism where half the sperm cell's descendants have the mutation and half of them don't. So this is kind of a violation of Mendel's laws, and here's an example of what that looks like in a certain organism. For instance, typical mold that you find on bread called Neurospora makes its spores. It gives one more cell division after meiosis in order to make the spores that will give rise to the next generation. So these heteroduplex DNA molecules can actually be different between these two different spores. And this is how we know that the products of meiosis can be different. And this is what those Neurospora spores actually look like. And it's been a really useful biological tool because at the end of meiosis, these 8 spores are all in a row. And if you take, let's say this row of 8, 1, 2, 3, 4, 5, 6, 7, well, maybe there's 9. There might be an extra in there. But if you have a nice ordered row of 8, you can tell that some of them came from meiosis 1. So with the meiosis 1, these 4 and these 4 were separate, and then meiosis 2, and then one mitosis after meiosis 2. So there's cases. If you see at the very end, if you see one that's different from its partner, then you know that was because there was this heteroduplex DNA that was only revealed after that mitosis. Okay, so that's kind of a lot of detail on that process of holiday junction resolution, and we can go into detail because biochemists have really figured out the whole pathway for these, and kind of in a lot of structural detail how these resolution events take place, and people can see the different kinds of joint molecules between different DNA molecules by kind of sorting them based on their shape. So that's we have a lot of molecular information on that, but I don't think it's useful to go into too much detail on the names of the proteins and showing how they do what they do. I think it's more important that we just get the concept. Mainly the difference between this picture and this picture, which superficially look the same, but the fact is that you, in order to get to this point where you have the crossover between two chromosomes, you have to start with this double, kind of like a double crossover, but it's not a double crossover. It's a double holiday junction between two DNA molecules. So any questions on that part you can ask now or later. For the last bit, I'm going to talk a little bit about not the structure of how this happens, but how what are the levels that these mechanisms are regulated on. So as I said many times, making a double-strand break on your chromosome is a highly dangerous event. And during meiosis, cells have evolved a lot of ways to keep control of this event so it doesn't get out, so it doesn't lead to death, basically. course, if you have too many double-strand breaks, you will just shred your entire genome into pieces, and you'll lose everything. The repair machinery is not good enough to handle thousands or tens of thousands of double-strand breaks. Actually, the typical numbers are just a few hundred or a few tens to a few hundred. So there needs to be ways to limit the number of double-strand breaks. On the other hand, if you do not make a double-strand break on your chromosome, then you will also lose that chromosome because it won't be able to segregate properly in meiosis. So there are different levels of control of these processes that I just want to go over these 4 levels here. First one is just kind of, it doesn't have a whole lot of consequence for for how meiosis ends up. It's not that biologically relevant, but it is relevant for people doing genetics. And that is that along an entire chromosome, there's actually a bias in where your crossovers will occur. So we know, for instance, that you can map genes. We've gone over this. You can have, if you look at outcomes of these kinds of crosses, and if you cross these together, and if you see that, for instance, RY is, say, 3 times less than small y and big P, that is just a direct reflection of the fact that this interval is 3 times smaller. So we can say maybe this interval has a relative size of 1, and this interval has a relative size of 3. But this, when you measure intervals like this, you're measuring what are called genetic distances. So these genes have a location on this virtual genetic map that is only measured by looking at recombination, by looking at the differences in offspring, for instance. But remember, this genetic map is this idealized line where genes are sitting, and they have a certain distance between each gene. So maybe that distance is 2. That's different from, so we can say that this is genetic length. But of course, the chromosome is also a physical object, right? It's got DNA, and we know now, because what we just looked at, that there's some propensity for recombination. So maybe you're getting some recombination in this interval. And what people have found is that the genomic length or physical length is another way of saying that. These are not the same. Okay, so that's the point. The distances on the genetic map, let's say there's a distance of 1 between R and Y, and a distance of 3 between Y and P. Those may not necessarily correspond to differences in the physical map. It could be that actually, if you look at where's the R gene on the chromosome, it might be here, but then the Y and the P gene might literally be right next to each other on the chromosome. So what could explain this? How could you have differences in the genetic and the physical map? Well, what this logically has to entail is that the probability of getting a double-strand break or a crossover is not the same for every single piece of DNA along the chromosome. And we know that that's definitely true now. And can anybody think of one thing that might influence whether you get this double-strand breaks on a chromosome? Something we talked about, and we might be able to connect.
The location where the skull 11 tend to bind to.
that definitely is the, I guess you could say that's the most proximal determinant, because yes, the SPO11 is not going to be equally likely to bind everywhere. And so, yeah. But then what might make that? Well, let's just leave it at that, because that, yeah, it turns out there's many things that influence where SPO11 might want to bind. Doesn't seem to be sequence specific. So the DNA sequence, of course, could be different here. There might be a certain sequence that really likes to get crossovers or recombination events. But things like euchromatin and heterochromatin also really affect this. So you're much less likely to get breaks in heterochromatin. So two genes that are separated by some heterochromatin, even though they may be very close together, there may be this region where SPO11 does not like to bind, or even if SPO11 does bind, maybe for whatever other reason, crossovers are not likely to happen. Then you will see a large genetic distance between these genes, even though, so yeah, you see this genetic distance, even though the physical distance is small. Likewise, there are other parts of the chromosome that are called hotspots. And that's where SPO11 really, really likes to bind. So it could be that, for instance, that would actually be here. So SPO11 would really like to bind in here. So even though the distance physically is small, since there would be so much recombination happening here, it would appear genetically to be very large. And yeah, it's things like histone modifications can affect that. Recently in mammals, it's been shown that there is actually some sequence specificity, because there's a protein that is not in all mammals, but it's in humans and mice. It's called PRDM9. It comes up sometimes in health news and things like that, because people who have different forms of this PRDM9 may have difficulties in fertility and things like that. This PRDM9 protein has different propensities, different probabilities of binding to different locations along the chromosome, and this tends to influence also where SPO11 will be binding and cutting. Anyway, the point is that along the chromosome, if you think of here's the entire chromosome laid out, there's what you might call this DSB landscape, where thanks to things like DNA packaging, heterochromatin or euchromatin, histone modifications, and then where's PRDM9 binding and where's SPO11 is binding, you can have some quite different probabilities of getting crossover somewhere. So you can have some cold spots where you'll never get a crossover. You can have regions where you're globally larger, and you can have some regions that are very, very likely to get a crossover. So the physical map being like this makes the genetic map kind of distorted, but if you only study one or the other, you would never know that there's any distortion going on. It's only when you try to combine the two that you see, wow, these genes with a large genetic distance are actually right next to each other, and things like that. Okay, so this is what you could call the crossover landscape, or the double-strand break landscape. They're of course related to each other, but there are some things that affect one and not the other. Okay. Yeah. Another category of this that's very important is homeostasis in double-strand breaks. So this is kind of, I just alluded to it a little bit earlier, that a cell needs to get a certain number of breaks on its chromosomes in order to be satisfied that meiosis can proceed. And so there was a really nice experiment where this SPO11 protein, normally we have 2 copies of every gene, and that means we have a certain amount of protein made. This really nice experiment, they kind of broke SPO11 so that they made some mice that had a really small amount of SPO11, some mice that had a medium amount, and then some mice that had a huge amount of SCO11, and they decided to see what effect does this have on the double-strand breaks. But in fact, it didn't really matter that much. No matter how much SCO11 there was, they still made just about the same number of double-strand breaks. So that means that, in some sense, the cell is kind of waiting, delaying the cell cycle until a certain number of double-strand breaks are made, which means that it's not just, the cell isn't just making double-strand breaks for a while and then moving on. Rather, this process is monitored so that the mechanism still isn't really well understood. But again, the number is about 200 to 300 double-strand breaks are finally made in the entire nucleus, and that seems to be enough to move on with the cell cycle. Just this, the cell isn't just blindly going ahead and making breaks all over the chromosomes, but it's watching very carefully how many it's making. So the other part of this that it was thought that maybe a mechanism like that could explain is the fact that if you look at the end of meiosis, every chromosome will always have one crossover, at least one crossover on it. And that's the next section of double-strand break or crossover control. There's this phenomenon called the obligate crossover, where, let's see, so the consequence of not having a crossover is, of course, you may lose the chromosome because, let's just draw this thing again. Because the crossover turns into the chiasma, which is the place where chromosomes are held together, right? So let's say the centromere is here. So the chromosomes are being pulled apart like this, and because they've done this crossover, or they just kind of turn this one around and twist the arms out, it can just turn into this shape. Let's see, the centromeres here. So the centromeres are getting pulled, and it pulls the chromosome into this shape. And because of the cystochromatic cohesion, all of these four DNA molecules, all these four chromosomes are held together and are able to support this tension from the spindle. And they can only do this, they're only connected because they've made this crossover. So if you didn't get this crossover, then all you have is, at the end, you've got two chromosomes with two centromeres on them. And they can both go that way, or they can both go this way, or randomly they could go correctly in opposite directions. That means you have a 50-50 chance of these two chromosomes going the same way, which is, of course, bad. You will end up with the aneuploidy, too many or too few chromosomes. All right, but if you are just making double-strand breaks randomly throughout your genome, and you have, let's say in the case of humans, we've got 23 pairs of chromosomes. I'm not going to draw all 23, but let's just draw a few. This will represent human chromosomes. And we're making double-strand breaks, and we're just randomly breaking the genome and doing crossovers. How many double-strand breaks would we need to make sure each chromosome gets a crossover? So if you're the cell, and remember I said just in the last step that there has to be this homeostasis, we were trying to make a certain number of breaks, then how many breaks should we make? So one break is obviously not enough because you'll just make one. And then two is not going to be enough because, again, you obviously need at least 3 breaks. But your third break might be there instead if you're just making them randomly. So you haven't made a break here yet, so maybe 4 is better, right? So let's make 4, but no, you still missed it. So the question really becomes, if each chromosome has an equal chance of getting a break on average, how many breaks would you need to make to be, let's say, 99.9% sure that each chromosome will get a single break? And that number turns out to be, for large numbers of chromosomes like us, that's really a lot. That's really a lot of breaks. It's hundreds and hundreds. And then you're still not ever 100% sure, right? And plus, that also means that perhaps one chromosome, if you have to make, let's say you had to make 500 breaks randomly for that to happen, one chromosome might get a huge number of breaks in one place, and that would not be good either. So at the end of it, there is actually it seems to be, that's not the way, so this is not the strategy that cells have. They don't have this, they don't have a strategy that's just keep making breaks and keep breaking and breaking until we think there's enough. There is actually a feedback mechanism so that chromosomes are monitoring whether or not they've had a break, or whether or not they've had a crossover. And it's thought that it's very similar to the mechanism that, for instance, mitotic chromosomes use to make sure that they're on the spindle or not. So remember, for that to happen, each unattached chromosome is sending some signal to the cell and saying, wait and don't divide yet until I'm attached. There seems to be a similar signal happening in meiosis, where each chromosome that doesn't have a crossover yet is also sending a signal that says, don't go on until I have got my crossover. We're still not sure exactly what the nature of that signal is, but in the end, this kind of mechanism does operate, and it lets the cell, it's a good thing that this happens, because it lets the cell get away with making fewer double-strand breaks than it would otherwise need to. You can say, we're only going to make 50 double-strand breaks, plus we're going to keep making breaks until every chromosome has at least one. You can get away with much fewer breaks that way than just making enough so that you're automatically 99% sure that every chromosome has to have one. Yeah, we can go over the actual math behind that if you want, but it is a little bit tricky. Right, and then the last one is kind of the most interesting one for me, because it's one that we're studying. And that is the fact that crossovers actually communicate with each other, and they don't occur randomly on the chromosome. And this is another way where you can avoid getting too many breaks or too many crossovers on a single chromosome. If you were just making a lot of breaks and turning them all into crossovers, then you would just be making crossovers between chromosomes, so many that it kind of defeats the purpose of having chromosomes, and that would be really hard to disentangle all that during cell division. So it seems that almost all organisms have this phenomenon of interference that has to do with chromosomes. If you get a crossover somewhere on a chromosome, that tends to inhibit or make less likely the future crossovers on the same chromosome. And so that was found when in another typical 3-point cross, and all of them use the same big R, big Y, big P, little r, little y, little p again. So when you're counting the kind of crossovers you can get this kind of crossing over between R and Y in these two ways. You can get crossing over between Y and P in these two ways. And so let's just, and these are going to be the same, so let's just combine them. And you can get triple crossovers. And those should also be the same. And at first, when people were mapping these things, it was thought that whatever probability there was in getting crossover between R and Y, let's say this is a 30% chance. And then let's say between Y and P, let's say you have a 50% chance. So half the time, that's a little too high actually. Let's make this 10%. So if these were the numbers you had, it was naively thought that, well, if you have a 30% chance of crossover here and a 10% chance of a crossover here, then getting a crossover here and here should be what?
3.
Mm-hmm. Yeah, if they're independent. But this phenomenon of interference just shows that these are actually not independent. And in practice, you get much less than 3%. It depends on an organism, and it depends on the distance between these. But in fact, the number of expected double crossovers you get is almost always much less. Sorry, the number of actual double crossovers you get is almost always less than the number of expected by just looking in single intervals. And it's not really clear, I guess, that people think why this might happen is just because you don't want to have too many crossovers right next to each other, because resolving all these, each crossover, of course, is a double holiday junction, and having too many double holiday junctions right next to each other in the same molecules may be a little too confusing. But in fact, interference can operate over much longer distances than would be than you would think would interfere with these double holiday junctions. So it could be more kind of like evolutionary reason where you don't want to shuffle the genes too much. So you prefer to have large blocks of chromatin that is not recombining with each other and passing these down. Rather, you want to recombine in just a few places on each chromosome. I'm not exactly sure about I'm not an expert in evolution, but I think it's likely that if you already exist as an organism, you probably already have a pretty good combination of genes. So you don't want to just mess everything up and completely shuffle your mother and father's genome, but rather you want to keep blocks together. So this might be one reason why crossovers are spread out like this. The last part I'll talk about. So interference can vary among organisms. It can be zero. So some organisms actually, like fission yeast, is one popular organism that doesn't seem to have any interference. You can have as many crossovers as you want on a chromosome. It doesn't matter. But the other extreme, where you have complete interference, is C. elegans, and that's why I'm very interested in it. So in C. elegans, let's see. Yeah, genetic interference is 100%, which means if you have crossover between R and Y, you simply will not have a crossover between Y and P on the same chromosome. Never. So in other words, R, Y, P will not be 3%, it will be 0% no matter what. So what does that mean? That means, so number of crossovers per chromosome. So there's still this obligate crossover. So every chromosome gets at least one crossover, but it doesn't get more than one crossover. In other words, 1. So C. elegans has 6 chromosomes, and in each meiosis, each chromosome gets only one single crossover, and only one. Only in certain mutant conditions do you ever have more than one crossover. And the last part would be just some speculation. We don't exactly know why that is, but it may have something to do with the fact that C. elegans chromosomes are holocentric, which means as opposed to us and mice and most organisms that have an actual centromere. I think I mentioned before, C. elegans has these chromosomes that simply, in mitosis anyway, the entire chromosome acts as a kinetophore. So the entire chromosome can nucleate microtubules and get pulled apart. But in meiosis, of course, if the entire chromosome could nucleate microtubules, if microtubules grabbed everywhere on the chromosome, then it would actually pull these arms apart as well. So actually in meiosis, even though C. elegans does have holocentric chromosomes, in meiosis it acts like a monocentric chromosome and prevents microtubules from attaching near the center of this, near the mitotic plane, near the antimetaphase plate. The mechanisms for how it does that are kind of a little bit advanced, but the fact is that if part of the mechanisms by which it prevents microtubules from binding here has to do with where the crossover is on the chromosome. And this kind of ties everything together, so it's maybe good to go over it. So C. elegans only has one and only one crossover on each chromosome at each meiosis. And because of this effect of double-strand break landscape that I talked about earlier, the C. elegans double-strand break landscape is actually pretty interesting. It looks like this. So C. elegans double-strand break landscape looks like this. If this is the left of the chromosome and this is the right of the chromosome. It's very likely that it will get a crossover either somewhere on the left or somewhere on the right, but it will not get a crossover in the middle of the chromosome. It has to do with the same kind of factors that affect that in us, so histone modifications and where SPO11 likes to bind and things like that. But the effect is that chromosomes get a single crossover, and it's either, here I'm showing top and bottom, not left and right, but it's either at one end or the other of the chromosome. But C. elegans is really, really interesting. It somehow can measure the distance from the crossover to the nearest end. So from the crossover, it says, okay, this is a short way to go, and this is a long way to go. And what C. elegans always does is it turns the long end, the long direction, into this microtubule nucleating end. And the short end is the end that's going to lose cohesion in the first meiotic division. Right? So let's contrast that with a typical, like you and me, our chromosomes. We always have a centromere. So the centromere is always where microtubules bind to. So the microtubules come out of here. So that's very easy. It's always here. And we always remove chromatid cohesion from this area in the first meiotic division. So pretty much everywhere except the centromere loses cohesion in meiosis 1. Did I indicate that? Yeah, sorry. This inner box here will be meiosis 1, where we lose cohesion, and this outer box here is meiosis 2. C. elegans has the same thing. It has to lose cohesion on part of the chromosome in meiosis 1, and it loses cohesion on the rest of it in meiosis 2. But that's determined for every chromosome independently, because it is based on where the crossover happens. So if the crossover happened near the left arm, then the left arms get put to the metaphase plate, and the left arms are where cohesion is lost first, and cohesion is maintained on these long right arms. But since this double-strand break landscape is like this, you're equally likely to get a crossover on the right arm. Then everything could be switched, and now if the crossover were over here, now these long arms will be the left end of the chromosome, and the right arms would lose cohesion first. So unlike our chromosomes that always do it the same way every time, C. elegans chromosomes are able to change where they lose cohesion and where is the effective centromere based on the position of the crossover. And so, yeah, it's still a mystery, but somehow they're able to measure the distance. Each chromosome is able to detect this is the short distance, so this is the shortest distance to the end, and this is the longest distance to the end, and then make something different happen on each part of this chromosome depending on the length. And that's another thing that my laboratory is investigating, but we still don't know the actual mechanism of how it's able to measure length like that. So yeah, I think that's pretty interesting, and I think that's where we'll end for today. Okay.